The first speaker of this session is Dr. Trevor Fidler. Dr. Trevor Fidler obtained his PhD at the University of Utah under the mentorship of Dr. Dale Abel. He did postdoctoral training at Columbia University with Dr. Alan Toll, where he identified the mechanisms by which clonal hematopoiesis promotes cardiovascular disease. Last year in the summer, he joined the faculty in the Cardiovascular Research Institute at UCSF. Today's his talk is title is Mechanisms of Atherogenesis in Clonal Hematopoiesis. The floor is yours.
Thanks. I'd like to thank the organizers for inviting me to talk today, and thank you guys. We had a little shift in the schedule. My son got sick, so I have to pick him up a little bit later. But I'm glad I can share this story today. So my lab started at the Cardiovascular Institute this summer, and we're interested in atherosclerosis and multiple aspects of it. But one of our major focuses is understanding how patients with why patients with clonal hematopoiesis have increased cardiovascular disease. So clonal hematopoiesis occurs when somatic mutations occur in your hematopoietic stem cells in your bone marrow. And then these stem cells, you can see in purple, give rise to mutated monocytes and neutrophils and other cells within the blood. And then over time or following stress, these clones can expand and providing a larger fraction within your blood. In patients, this is usually represented around 2 to 15% of the blood cells carry these mutations. So clonal hematopoiesis, although they're cancer mutations, really only about 1% of these patients will transform into a cancer or hematological malignancy. But clonal hematopoiesis is associated with a 40% increase in death. So the mutations that give rise to clonal hematopoiesis, or I may call it CHIP, clonal hematopoiesis of indeterminate potential, which it's all the same thing. This can occur in a variety of genes. So primarily it occurs in DNA methylation enzymes responsible for putting methylation or methyl onto DNA or off with DNMT3A as well as TET2. Also modifiers of histone function or DNA damage repair with PPM1D or P53, as well as cell signaling like JAK2, which I'll talk about today. So there's a growing body of literature really demonstrating that if you have clonal hematopoiesis driven by these different mutations, they lead to different outcomes in patients. So to help explain the increase in mortality, Sid Jaiswal down at Stanford now and Ben Ebert had a seminal report showing that patients with DNMT3A, TET2, ASXL1, or JAK2-driven clonal hematopoiesis have increased coronary artery disease. You can see these odd hazard ratios here. Overall, it's close to 2% of an increase. So to investigate causality, Jose Fuster and Ken Walsh modeled TET2 clonal hematopoiesis in mice. They then examined atherosclerosis. So you can see here that the lesions from mice, which are marked as 10% knockout, these have 10% of TET2 knockout cells, which mimic the mutation, have increased atherosclerotic lesion area. However, when they treat with MCC950, which is an NLRP3 inflammasome inhibitor, they were able to reduce this increase in lesion area, establishing a link between clonal hematopoiesis and inflammation. So the inflammasome, as Mikey kind of talked about earlier, has emerged as a major contributor in cardiovascular disease. So primarily, the inflammasome can be driven by NLRP3, which can be activated by various ligands, including cholesterol, oxidized DNA, and others. And the AIM2 inflammasome, which really specifically binds to double-strand DNA breaks. Upon activation in the cell, these activate caspase-1, which proteolytically cleaves pro-IL-1 beta into the active form, which can then be released when the cell also cleaves gasdermin D to form the pore in the plasma membrane. And these are both facilitated by caspase-1. So IL-1 beta has been shown to be beneficial in patients, and just real quickly, these data are probably familiar to everybody here, is that patients given canakinumab antibodies to IL-1 beta had reduced cardiovascular disease relative to placebo in the CANTOS trial. And then after the CANTOS trial, a subset of patients were sequenced, and they found that patients who have TET2 clonal hematopoiesis actually did even better when you give them IL-1 beta antibodies, kind of suggesting that, in fact, the pathology is driven by IL-1. This was a very underpowered study, so it's hard to draw too many conclusions, but the human data were there. So during my postdoc, we became interested in JAK2, and this was because JAK2 had almost a 12-fold increased hazard ratio for coronary heart disease. JAK2, also these mutations are in the V617F, and they lead to a gain of function. And JAK2 is highly prevalent, so it's found in about 3% of just a general European population. So my initial work on this topic dissected the mechanism by which JAK2 mutations in macrophages may promote atherosclerosis. So this work demonstrated that these JAK2 mutations act in conjunction with MCSF, so macrophage growth factors, to promote proliferation with IL-1 beta signaling. And this led to increased reactive oxygen species and DNA replicative stress within the macrophages in the atheroma. This then promoted the macrophage accumulation through increased proliferation, as well as through increased recruitment. And then once the mutated macrophage is accumulated in the plaques, the DNA damage that occurred from the increased proliferation activated the AIM2 inflammasome, releasing more IL-1 beta to kind of perpetuate the cycle. And these cells ended up dying through pyroptosis and increased the necrotic cores in the lesions of future plaque instability. So from that work, we decided to look at fibrous cap thickening in these mice, because we found that really on 4 different methods, deleting the inflammasome in hematopoietic cells led to an increase in cap thickness marked by caspase 111 deletion or gasdermin D deletion, as well as pharmacological inhibition of IL-1 alpha and beta with anakinra, and antibodies to IL-1 beta from Novartis that all led to increased fibrous cap thickening, and we saw no impact in the controls. So fibrous cap thickening is one of our best correlates to human plaque stability, and so we were quite interested in this. Another important point was that we did not find any effect in LDLR mice or the hypercholesterolemic mice without mutations in JAK2. So we really didn't find prominent inflammasome activation, very little benefit unless you have prominent inflammation. So this work was just published this month, and working with Lars Magnefessel at the Munich Vascular Biobank, they did laser capture catapult of the fibrous cap in stable and unstable atheromas from patients. And then we did qPCR analysis of the fibrous cap region, and we found there was, in fact, more IL-1 beta expressed within the cap. Then, using single-cell RNA sequencing of these atheromas as well, we looked for which players might be expressing IL-1 beta and which might be receiving the signal. So we found that IL-1 beta and components of the inflammasome were primarily expressed in inflammatory macrophages, monocytes. You can see a little bit here, whereas very little IL-1 beta was expressed in the fibroblasts, endothelial cells, or the smooth muscle cells. Importantly, IL-1R1, which is the primary IL-1 receptor, was really primarily just expressed in endothelial cells, smooth muscle cells, and fibroblasts, and just a little bit in the hematopoietic cells. So based on those data, we wanted to see, well, which cells were the macrophages signaling to? So we utilized these My11 tamoxifen-inducible ZS green reporter mice. So essentially with this model, we're able to find all of the smooth muscle cell-derived cells using a ZS green reporter. We transplanted either JAK2 or TET2 bone marrow, which models the clonal hematopoiesis in patients, into these reporter mice, gave them atherosclerosis, and then at the end of the study, we gave them IL-1 beta antibodies or IgG control as a therapeutic. We then, or to model the therapeutic, we then sorted the cells for ZS green positive, so smooth muscle cell derived or non-smooth muscle cell derived, and did single cell RNA sequencing. So this approach in the mice with JAK2 clonal, or modeling JAK2 clonal hematopoiesis led to about 18 different populations, which were primarily smooth muscle cells, fibroblasts, macrophages, a little bit of endothelial cells. But we were surprised to find that when we looked in the smooth muscle cell-derived cells, the ZS green positive cells, we really found very little changes in the distribution of smooth muscle cells. I apologize for the very generic titles of the populations, but really in general, we find very little change in the smooth muscle cells. Cluster 5 does expand a little bit with IL-1 beta antibody treatment, and those are contractile looking. However, we were happy to find that TREM2 macrophages decreased, which was similar to previous data we had published and kind of consistent with IL-1 beta antibodies reducing immune cell infiltration into the plaque. But we were surprised to find about a twofold increase in these fibroblast-like cells, which were not derived from smooth muscle cells. Interestingly, within those smooth fibroblast-like cells, those cluster 6 fibroblasts, they were highly enriched for matrix proteins, COL1A1, COL1A2, lubricin, decrin. These were really the most enriched genes within these clusters, suggesting maybe they could be helping build the fibrous cap. We did the same analysis in mice modeling TET2 clonal hematopoiesis, and we find about 21 populations, and again, the distribution is somewhat similar, fibroblasts, smooth muscle cells, and myeloid cells. And again, just like in the JAK2 model, we find no expansion of major smooth muscle cell populations. However, we do find an expansion of these cluster 0 fibroblast-like cells. I think it goes from about 14 to maybe 20% of the populations. And again, these are not derived from smooth muscle cells. And importantly, the clusters that were most enriched in this population were just like the cluster 6 fibroblasts from the JAK2 model, suggesting that in 2 different models of clonal hematopoiesis, IL-1 beta inhibition expands a non-smooth muscle cell-derived population. So to look at this a little bit closer, we wanted to find a histological marker so we could get past the single-cell approach. And we found that decrin, which is a proteoglycan, was really only expressed in the ZS green negative population. So this is just single cell of everything in bulk. And you can see decrin is highly expressed here. And then when you look within the clusters, you find decrin enriched in the fibroblast-like cells, although it's not 100%, but it's close. So when we look in the atheromas, we stain for decrin in red, and you can see that in mice treated with IgG on the left. And you can see that on the right as well with mice treated with IL-1 beta antibodies. So the ZS green are all the SMC, or the smooth muscle cell-derived cells as well. And you can see these dashed line represent the media. You can see there's a lot of decrin-positive cells within the adventitia. And you can kind of see some peppered here within the media and maybe kind of crawling along the media, traversing this acellular region and really becoming enriched within the cap region. So, this is this is a snapshot, but kind of the running interpretation of this is that the fibroblasts probably come from the adventitia, migrate through the media into the plaque, and then help stabilize the plaque by ending up in the cap. And also just fluorescence intensity of the protein was also significantly increased. So to try to get at what these fibroblast-like cells might be doing in the plaque, we looked at PRG4. This is another proteoglycan. We were interested in it for a few other reasons, but we found that there was some enrichment in fibroblast-like cells. And following administration of anakinra using RNA-scope, we found that, in fact, PRG4 positive cells increase within the intima, and those are marked in red here. So we utilized this somewhat complex model where we did a PRG4 Cre-driven diphtheria toxin A mouse model. So when we gave these tamoxifen, we could deplete all the PRG4 positive cells. We modeled JAK2 clonal hematopoiesis in these mice, gave them a hypercholesterolemic diet for 10 weeks, and then we gave them IL-1 beta antibodies. And we found that in mice modeling JAK2 clonal hematopoiesis, there was no significant increase in cap thickness. There was actually a reduction. And in wild-type mice, or mice with wild-type bone marrow, not JAK2 bone marrow, we don't see a significant reduction, but we do see a trend down. So this is consistent with the idea that you can only benefit from IL-1 beta therapy when you have prominent inflammation. I was a little ambitious, so I'll try to go through this section very quickly. So the next question is, we were looking at JAK2 mutations, and there are JAK inhibitors. So we asked the question, will ruxolitinib, which is a JAK1/2 inhibitor, increase cap thickness and plaque stability in mice modeling JAK2 clonal hematopoiesis? So we did this, and we were really surprised to find there was a significant reduction in the lesion area, but they were super necrotic. They were very unhealthy looking. And so, these acellular regions may occur in unstable plaques. And also, the fibrous cap was significantly reduced. This was really, we were pretty surprised by this. We didn't expect a bad result. But then a few months later, the FDA actually put out a warning for tofacitinib, which is a JAK inhibitor as well, which is used in rheumatoid arthritis. And this warning was for heart-related events. And it's not quite clear exactly what's happening here. But these patients with rheumatoid arthritis taking the JAK inhibitor are having heart events or issues. You can see the increased hazard ratio. So working with Ross Levine and Andy Dunbar at Memorial Sloan Kettering, we asked, well, can we selectively inhibit the JAK2 mutation? So kind of model what it would be like to develop a JAK2 selective inhibitor, a mutant selective inhibitor. So we utilized these mice using a DRE-CRE system. So essentially, the mice express the JAK2 mutation. Using DRE recombination, you can express in the right orientation, by recombining the ROX sites. And then we have a tamoxifen-inducible CRE in the same mice. So then, whenever we want, we can turn off the JAK2 mutation by just treating with tamoxifen, and the LOX-P sites will cut out the mutated exon. So you can see here that after 5 days, in fact, the blood monocytes, which are elevated in mice modeling JAK2 clonal hematopoiesis, come back down to a normal control level. Also, at the end of the study, you can see the spleen weights go down. This is a major feature of the JAK2 mutations. However, one complication was when we we turned off the JAK2 mutation with tamoxifen, we actually get an elevation in serum cholesterol. And this is problematic because cholesterol promotes atherosclerosis. But this increase in cholesterol, or plasma cholesterol, also happens when you give ruxolitinib. So we asked, well, maybe in conjunction with cholesterol lowering, inhibition of JAK2 mutations will improve atherosclerosis. So we did a new cohort of mice, and what we did is we gave the mice atherosclerosis. We put them on a Western-type diet for 10 weeks, and then we gave them LDLR virus into their liver, which restores LDLR expression and then allows you to lower cholesterol. So by doing this, we were able to control for that increase in cholesterol levels and bring them down. And this was a model of plaque regression. This would be similar to patients taking statins. So when we looked at the lesions of these mice, we found that, in fact, in conjunction to cholesterol lowering, we were able to increase the fibrous cap thickness, a major feature of plaque stability, by turning JAK2 mutations off. And we also, marked in magenta here, we also found that there was an accumulation of Decrin-positive cells within the intima, or within the atheroma, suggesting that potentially this is also leading to an expansion of these fibroblast-like cells in the plaque. So now we're getting to the end. So to summarize, selectively targeting JAK2 mutations in combination with cholesterol lowering may reduce cardiovascular risk in patients with JAK2 clonal hematopoiesis. And kind of all together from the first part, and the part I didn't show too much about, but we found that JAK2 mutations in macrophages promote AIM2 inflammasome activation, IL-1 beta secretion, and increased proliferation of macrophages, as well as increased recruitment of monocytes into the lesions. It also leads to increased pyroptosis in the necrotic core area. And when we inhibit IL-1 beta, we're able to bring fibroblasts into the plaque, which may lay down the matrix to make them more stable. And we think that this might be an explanation why Canto's trial was successful, because actually in a subset of patients treated with IL-1 beta antibodies, they did imaging on these people, and they didn't have any change in their lesion area, but the benefit of IL-1 beta on atherosclerosis has been established now. So then just the last piece of data, all of the work I showed you today was really in mice and some human correlates, but in collaboration with ZU in Pradeep Nigerian's lab at the Broad, we looked at the UK Biobank, about 425,000 patients. And we asked the question, well, if you have JAK2 clonal hematopoiesis and increase AIM2 expression, which is predicted off their DNA SNPs, do you have increased cardiovascular disease? And in fact, we did find that to be the case. So cardiovascular disease is the hazard ratio on the bottom. And you can see that patients with JAK2 mutations and increased AIM2 expression have increased risk. This was not true for TET2, whose inflammasome activation has been shown to be through NLRP3, or DNMT3A. However, we do find that with ASXL1, as well as if you have low IL-10, you also seem to be protected. So in that same report, we just kind of showed that they also have AIM2 inflammasome activation in ASXL1 mutants, and IL-10 secretion is up. This is kind of a future direction of the lab that we're working on, trying to understand why you have increased IL-1, increased IL-10, and increased athero potentially. So just from all that, I'd like to thank people at Columbia who did most of this work with me. My postdoc mentor, Al Tull, Murdoch Riley did a lot of the smooth muscle cell tracing with us. Peter Libby, Pradeep, and Z for their work with the genomics. We didn't talk about the work with Ben. And Ross and Andy for their work with the DREAM mice. And Lars and his team in Munich for their work with the human atheromas. As well as now the lab is growing at UCSF. We have June Lau, who's here, and she's really spearheading a lot of the continuation of this work on her own at the moment. But hopefully we'll start filling up the lab a little bit further. And with that, I can take any questions.
Great talk. Any questions?
Great stuff, Trevor. I had two questions. One was on the fibroblast. You showed us two clusters, and one of them was not affected. And I wonder which kind of fibroblasts are those? Do you see difference in I1, R1 expression in these two clusters that are reactive or non-reactive? And then the second one is that you have a pretty dramatic expansion of the CD8 T cells, and I wonder if you gave any thought on that.
Yeah. The first question about the fibroblasts impact of IL-1 on them. So we did differential gene expression across the board on pretty much every cluster we had in mice treated with IL-1 beta antibodies and mice with IgG across clusters. And we really just couldn't find anything. We find like 8 genes that are differentially expressed across the 2 in vivo treatments. One interpretation I have is this is occurring at the progenitor level. So somehow we're hitting like a mesenchymal stem cell with IL-1. Now it's producing more fibroblasts, and they all look the same. I don't know if that's the case. We did a mesenchymal stem cell CRE with IL-1, R1 deletion, and it actually had no impact here. So I don't really know how IL-1 is particularly affecting the fibroblasts. I mean, one point I kind of skimmed over, but IL-1 is also reducing the amount of macrophages coming into the lesions. So I think it's quite possible that mutated macrophages are releasing something that might be suppressing the fibroblasts from coming in. And when we inhibit IL-1 beta, we're actually working to reduce immune cell infiltration, which is then allowing fibroblasts to come in through the absence of some sort of repression signal. Very complicated, but these are things I'm thinking about and that we're trying to start to look at on exactly how they're communicating. It may be a direct communication of IL-1, but it also is kind of weird because then once we take away the signal, they come in. So why would that be? Now, the CD8 cells, these have always been, they're prominent changes. We don't know what's going on. They're CD8. We don't know where they are. Histologically, we can't find them. We thought there might be atlos. We can't find any of these. We don't know where they are, but they're certainly up in maybe 4 different single-cell analyses. We find this. It's a consistent change, but histologically, I can't find them. So we'd love to do that. Yeah, Chad.
That's awesome, Trevor. I'm curious if you've been able to tease out in your single-cell data set receptor-ligand interaction, particularly through something like cell chat, to be able to think of downstream of IL-1 what might be mediating some of these kind of cell-cell interactions.
That's definitely been considered. I have not done that. I think that's a great idea to kind of dissect that further. As I was just kind of mentioning to Mikey, I mean, statistically, we find 8 significantly changed genes across IL-1 antibody treatment groups. But I guess the cell chat would really be more of a composite, right? Like where it would take into account sub-significant changes, I guess, to piece them together for a pathway change. Maybe that might come up. We've done like silly pathway analyses. I forget what they were exactly, but not cell chat. And we don't find anything too interesting. We find some like migration proteins, which we're trying to look at and knock down and put in the lab, see if they might be affected. But we haven't found a great signature of something that might be changed, just from kind of very basic analysis.
I just want to clarify, your mutation of JAK2VF is a gain of function mutation, right?
The JAK2 mutation is a gain of function.
So in my understanding, these CHIP mutations are kind of in the multiple hit theory that eventually make cancer cells. So you showed DNA damage in the mechanism of JAK2 as a kind of final impact. So does this DNA damage make a new somatic mutation?
So, yeah, they could. So it's been identified that they have replication fork stalling in these in hematopoietic stem cells from the cancer literature. Whether that then introduces somatic mutations which get carried over, potentially. Typically, when you sequence, when you look in UK Biobank or other large genetic analyses, you find people with clonal hematopoiesis who have 2 different mutations, and there are some that are inherited together pretty frequently. You know, ASXL1, TET2, things like that. But with JAK2, you don't actually find that with too many guys, although it's not too common. But typically when you have JAK2, it's a strong enough mutation that it can transform on its own if it wanted, I guess.
Thank you. Amit.
Yeah. Hi. I just have one quick question. So did you get a chance to look which cell types in your atherosclerosis plaque were Decorin positive?
Yeah, so using the violin plots, very kind of crude way, we don't find it in any smooth muscle cell-derived cell, but we do find it primarily in fibroblast-like cells. We don't see any in the macrophages. You know, the reality is it's a marker. It's not going to be 100%. Same thing with the PRG4 approach. Obviously, it's great. There's a lot of CREs at UCSF for fibroblasts that we're trying out. There's a lot of different approaches we'll try to take. I don't think we'll ever be 100% specific on them because it's kind of a composite of expression patterns. But the idea that fibroblasts are coming in was somewhat novel here, just that they're becoming protective here, whereas Mikey just gave a long talk about how they were so bad for the heart. But if they happen in your vessel, we're proposing that maybe it's actually beneficial. So we're kind of in the early days of trying to figure this out. I know a lot of work's going on at Stanford right now. We should touch base at some point and see if there's some synergy there. Yeah.
Thank you.
Any other questions? If not, we will move on to the next speaker.
Thank you.